what's happening in the brain of people with OCD, with bipolar disorder, with schizophrenia. I study the neurobiology of psychiatric disorders. I want to understand in very specific detail the pathophysiology is a fancy term for how the illness happens and works in the brain. I'm working on a molecular, biochemical, cellular, and systems level of analysis where I am directly manipulating the brains of preclinical translational animal models to reverse translate different contributions to different illnesses that have been found, namely genes. We have human data from people who donate their tissue while they're alive, and we're able to do genome-wide association studies to find in large groups of people genetic differences that happen to be in people with an illness versus without. I have been taking candidate risk genes of OCD, bipolar disorder, schizophrenia, and I'm looking at what those genes do in the brain to contribute to different symptoms and the experience of the illness. Psychiatric illnesses have many different genes that contribute to each of them. It's not like there's one OCD gene or one bipolar gene. It's probably in the several hundreds for each of them. But when we find different genes or groups of genes that are different in people with a certain condition, the question becomes, what are those genes doing? Because the whole point of genes is that they help shape your body. They're the instructions to tell your body how to build itself and operate, especially in the case of the brain. I'm taking these genes and I am directly manipulating them in very specific brain regions that are of interest in these conditions. We're looking at what does that gene do to brain regions, to properties of the cells, to different biochemical signaling pathways, and what is it doing differently that contributes to different behaviors, to different symptoms, and ultimately all the contributors to what we end up calling and identifying as a psychiatric illness. Also, I'm going into postmortem brains of people with these conditions who donate their brains. We're able to take different homogenates, meaning almost a super smoothie of those very specific brain regions, and we're able to look at them and analyze for different protein levels, which are often a result of genes doing their thing. So we're able to see if those different proteins, those biochemical signaling pathways, are altered in the same way in humans, thus validating that connection between what we're finding preclinically, what we're finding in humans. Maybe you're wondering, what is the point of all of this? Well, we have to understand what's going on in the brain of someone with this mental illness so that then we're able to intervene and develop novel therapeutics to fix or change exactly what we're seeing that's driving the illness. In an ideal world, we're developing precision therapeutics to be able to precisely modulate and change the brain activity in specific regions and cell types to be able to ultimately help people like myself living with these illnesses.